Okay.
All right, everyone. So this meeting is being recorded. I want to go ahead and get started so that we stay on track. And so I'll go ahead and start with our welcome, and a big welcome to all of you, and a huge thank you for you guys taking your time out of your day to tune in to the NET Cancer Foundation's 3rd annual virtual research conference. We're very fortunate to have a great lineup of speakers today. We're excited to hear from all of them. Some of them will be recorded and some of them will be here live. If we have time, we will accept questions after each speaker. We'll just see what the time looks like. And so again, thank you all for coming for our 3rd annual virtual NET Cancer Foundation conference. And I'm going to go and pass it over to Angaraj so he can introduce our first speaker.
Thanks, Rachel. So it's a pleasure for me to introduce Dr. Chrissie Thirwell, who's very well known in the neuroendocrine field. Chrissie was appointed as the Merrill Gillings Professor of Cancer Genomics at UEMS in August 2019, and Associate Dean of Clinical Relations in 2022. Until moving to Exeter, she was a senior lecturer and consultant medical oncologist at the UCL Cancer Institute and Royal Free Hospital, and led consultant in the multidisciplinary neuroendocrine and hepatocellular cancer teams. She has been serving in multiple consortiums related to the neuroendocrine tumor. She has an international reputation for research into and on the management of neuroendocrine tumors and integrated genomic analysis of cancer. She's secretary of UK NETS and ENETS advisory board member, and she's co-chair of NETORF Board of Scientific Advisors, which are the major neuroendocrine bodies in the US and the UK. Our research group has focused on integrated genomic and circulating free DNA analysis in neuroendocrine and other solid tumors. Without further ado, here's a talk from Dr. Thirwell on neuroendocrine tumors, cracking the epigenetic code.
Hello, my name's Chrissie Thirwell, and I'm really sorry that I can't be with you today to give this plenary talk for the Neuroendocrine Cancer Foundation. Over the next 20 minutes or so, I'll give you an overview of some of the work that my group's been doing over the last 10 years in looking at the genomic makeup of neuroendocrine tumours, with a particular focus on epigenetics and why that's really important in the development of this particular type of cancer. So what I'll be covering is an introduction to cancer epigenomics and what we can learn from studying the DNA and the genome of cancer, and then how we've been applying this in neuroendocrine cancer over the last 10 years or so. I'll touch on some of the challenges of when we're analysing bulk tumour tissue, because each of our cells has individual differences and changes in its DNA, but a lot of our work and research is looking at bulk tumour tissue. And then how most recently we've gone on to create a DNA methylation atlas, which can help undo some of those challenges around analysing bulk tumour. And then I'll touch on some future applications towards the end of the presentation. So I'm going to start with Radiomics 101. The human genome is a wonderful thing. We have 3 billion base pairs of DNA that measure just over a metre in length, packed up into each of our nucleuses in our cells, which measure about 6 microns across. Every time a cell divides, this is unpackaged, copied, packaged back up again. And as you might imagine, there can be mistakes and errors in this. And that's the foundation of a lot of the changes that we see in the creation of cancer. So sometimes cancer can be thought of as a disease in aging, because as cells turn over a certain number of times, there's a higher chance that errors can be made in replication, or there can be external factors such as smoking or sunlight, which can drive those mistakes to be made more quickly. But work in looking at the genome and our DNA has accelerated phenomenally over the last 10 years. Just the rate and pace in which we can look and examine our genome has really, really come on. We can actually now do work to sequence our 3 billion base pairs of DNA in about 72 hours. And when I did my PhD 15 years ago, it took me a year just to look at one gene in around 100 cancers. So that's the rate and pace at which our work has come on. And it's a phenomenally exciting area to work in. I'm going to give you an explanation of some definitions, which I'll be referring back to through the talk. So we sometimes talk about germline and somatic variants, and germline variants are DNA that's in all of our cells, and it's present in the egg and the sperm that makes us, and it can come from your mother or your father. And these changes in the DNA are inherited, and it's present in every cell in your body. And we can pass those changes on to our offspring when we have children. When we talk about somatic variants, and that's the majority of the work we do when we actually study DNA from cancers themselves, these changes in the DNA aren't there in the egg and the sperm, but they can change at any time during development. So even in the womb, through growing up, through childhood and into adulthood, you can have these changes in the DNA, which are then present and can be picked up and detected in cancers. And these changes aren't passed on to our offspring. So germline variants, inherited cancer, somatic variants, changes that we see in our cancers and can give us clues about where they've developed and how they've come about. There's an enormous amount we can learn by studying the cancer genome. And there's been a phenomenal body of work internationally over the last 15 years, where we now have atlases of many different types of cancers, of thousands of cancers where their genome's been sequenced, and researchers can access this, and you have that as a benchmark as to what the cancer you're studying might have in common with other cancers, and might even have differences in terms of other cancers of the same type. So we can learn about how the cancers develop, the biology. You can identify new drug targets. We can actually measure tumor DNA in the bloodstream, so that can tell us how much cancer is there. It can tell us if perhaps the patient is responding to treatment. It can tell us about how cancer spread, so that's metastasis. And further down the line, it can tell us how a cancer might then develop resistance to the drugs that we're using to treat that cancer. And when we think of neuroendocrine tumours and of the work that's been undertaken to date, we know that they're clinically and genetically a heterogeneous group of tumours. And when we're deciding what treatments to give patients, we use the histology, it's what we call the gold standard. So we still rely a lot on how a tumour looks down the microscope, rather than a lot of different molecular or DNA markers to guide our treatment. We know that most commonly they occur in the bowel, the lung, and the pancreas. And we use quite a crude marker of the cellular proliferation. So the Ki-67 tells us how many tumour cells are turning over at any one point. And so that, in combination with where the cancer arose or started in, gives us an idea of what treatment types we might start. So, for example, we know that pancreatic tumours often respond better than intestinal tumours to chemotherapy. That's just one small example. But we use this quite basic information to help navigate really complex treatment pathways. Alongside the pathology of tumours, we also know that a certain proportion of neuroendocrine tumours can be inherited. So these are the germline mutations that I mentioned. So towards the bottom here, there are heritable syndromes in genes such as menin, neurofibromatosis, NF1, and von Hippel-Lindau, VHL. And that accounts for around 10% of cases. And my group and many others have spent the last 10 years trying to understand the somatic changes in neuroendocrine tumours by studying their DNA. And what we do in our tumour board meetings is we take really quite crude information to help guide us through quite complex treatment algorithms. There's still a lot of work to be done as to which order of different treatments we use if the tumour is advanced or has spread to other parts of the body, but we will often use the primary site and the Ki-67 to help us really get through quite complicated algorithms. And what I hope, and what work of many others hopes, is we'll move towards using epigenomic profiling to really help us get a good idea of prognostication and what treatment to give at diagnosis, and then use more personalised treatment and management as a patient might go through the course of their cancer management. And it may be in the future we might use this blood-based circulating tumour DNA to work out resistance to treatment and then access novel therapeutic targets. So this is my utopia of where we're working towards based on the work that we've been doing. I'm just going to give an introduction to what epigenetics is. So I've told you about the DNA sequence of 3 billion base pairs in the genome, which when that's read, it makes proteins, and those proteins are our building blocks of everything that make us. And it's also the building blocks of what makes tumours and cancers. So epigenetics can be defined as a heritable change in gene expression, so what makes those building blocks without a change in the underlying DNA sequence. So these are modifications to our DNA here. So in this cartoon here, this is the sequence of the letters of the DNA, and it gets wrapped up and it gets condensed into the chromosome. So when our cells replicate, this all gets unpackaged, and this is really important machinery for unpackaging. But epigenetics have different marks, one of which is DNA methylation, where a methyl group's added to a cytosine base next to a guanine. That's called a CpG. There are histone modifications, acetylation and methylation, and these histones are really important for packaging up that DNA into the chromosomes, and then it's unpackaged when the DNA is replicated. There's also microRNAs and non-coding RNAs. But for the purposes of the rest of the presentation, I'll be talking mainly about DNA methylation and histone modifications. So DNA methylation is one of the best studied epigenetic modifications. Of our 3 billion base pairs, about 27 million of them have a methyl group. It's like a little lollipop stuck onto the DNA. And these can actually switch on and off genes. So they have physiological roles. It's important, for example, in X chromosome inactivation in women. We only express one of our X chromosomes, so physiological roles, but also it can go wrong. It can lead to disease and cancer development, so pathological as well. And we can use DNA methylation marks across the genome to tell us a lot about different types of cancer. And one way or another, my group has applied all of these approaches on this slide into neuroendocrine tumours, and I'll cover them as we go through the next 10-15 minutes or so. So we know that DNA methylation patterns are specific to different tissue types in the body. We know that the methylation patterns are specific to different cancer types. So because of that, we can use a methylation pattern to tell us the primary site of a tumour. Quite commonly in oncology, you might have found a tumour that has spread to the liver, for example. You can do a biopsy and it's not entirely sure where the primary site of that tumour was. It could have come from the bowel, it could have come from the pancreas, it could have come from the esophagus, there could be other places it could have come from. But the methylation patterns can help tell us what that unknown primary site is. And that's really important because it's absolutely vital to get the right treatment. So different types of cancer require different types of treatment. So we can use the patterns to identify unknown primaries. We also can look at the biology of tumours. So we have differentially methylated genes across normal and across different tumours, and that can tell us about the biology and how the cancer developed. It can give us an estimation of the age of tumours. I'll give a nice example of epigenetic clocks in NETs further in the talk. And we can also use the methylation markers to tell us what types of cells make up a cancer. And that's really, really important information about how a cancer might have developed. So I'll just go back. And finally, and something I believe is really, really important, is we integrate that methylation data with the gene expression. So we can actually look at the impact of changes in the DNA sequence, changes in the methylation, and integrate that with then what we call the expression, the RNA that then leads to these protein building blocks. I think it's really important to put all of that information together so you can really pull out the biology and what causes the cancers to develop. So one of the most common approaches in the lab to look at methylation across the genome are arrays. You might have been familiar with arrays for looking at gene expression analysis, but there's similar arrays for looking at DNA methylation analysis. And these figures here just give us some idea and a sort of an overview of the different patterns of methylation. So I've already explained that we know that normal tissue can have a specific methylation pattern, but normal lung tissue is different to normal bowel tissue, is different to normal types of pancreatic tissue. So you have to have the right matched normal tissue to then look at the cancer. So you can look at different methylation patterns between normal tissue and the cancer. You can look at different methylation patterns in patients who might have a really good long-term survival and in the same cancer type as a patient who might do less well. So that can give us ideas and information about the biology that drives longer term survival as opposed to poorer survival. We can use methylation patterns to tell us whether a tumour might respond better to a certain type of treatment or worse. And all of this information is really important and useful clinically. And something that my group's worked on, but I won't be presenting today, is we've used methylation patterns to understand and identify neuroendocrine tumours of unknown primary from different sites. So most commonly it's either intestinal or pancreas, but we've also included some lung in that. And moreover, we can use different samples and tissue types to run methylation analysis on. So of course we can look at tissue, so tumour tissue, but also in saliva, blood, stool, urine. So there's lots of other biological sample types that we can use to study. So there's been concerted effort over the last 12 years to really understand the genomic makeup of neuroendocrine tumours. This has been through large-scale collaborative international work, and my group was involved in the work in the small intestinal neuroendocrine neoplasms here, where 180 tumours were sequenced. Very few mutations were found, and only mutations were found in 8% of cases in this gene CDKN1B, which is what we call a classic tumour suppressor gene. It's part of the cell cycle. Moreover, if a tumour harboured a mutation in that gene, it didn't give us any information on whether that tumour was more aggressive, less aggressive, or didn't link in with the outcome for that patient. So as an oncologist and a cancer doctor, this work didn't really take us very much further forward in how we might use the DNA makeup to target treatments or learn about prognosis. In pancreatic neuroendocrine neoplasms and bronchial neuroendocrine neoplasms, the story is a bit different. In around 40% of cases, there's mutations in that epigenetic machinery. So I highlighted those histone modifiers and chromatin remodelers when the DNA is condensed into chromosomes. So these are really important bits of our epigenetic machinery. So commonly in HRX and DAX and menin, MEN1. And there's also a large-scale piece of work of whole genome sequencing in 102 pancreatic neuroendocrine neoplasms by Aldo Scarpa's group, which identified mutations in DNA repair genes, so MYH and BRCA2, and also in the mTOR pathway. So this is kind of a concerted piece of international work over the last 10 years. And the bottom line is, and the most challenging thing for us in the research and clinical research community, is there's no common actionable mutations in this tumour type. And what we mean by actionable mutations are mutations where we can then use a molecularly targeted drug. So for example, in bowel cancer, you might use a drug against EGFR. In melanoma, you might use a drug against BRAF. So that's been one of the most surprising and challenging findings for us when we've been doing the genomic sequencing in recent years. The other really interesting observation is that neuroendocrine tumours have one of the lowest background mutation rates of all solid cancers. So this data comes from one of the atlases I mentioned, the Cancer Genome Atlas, and this is a logarithmic scale. So on the right here, you have very high rates of background mutations in lung because of smoking and melanoma because of UV damage. But neuroendocrine neoplasms come in here, one of the lowest background mutation rates of all solid tumours. And I think that tells us something very significant about the biology of these cancers. But we haven't really quite cracked exactly what that means, but it's just a really interesting observation at this stage. Over the next 5 minutes or so, I'm going to give you some insight into some of the work we've been doing in intestinal neuroendocrine tumours. Most of the work over the next few slides has been undertaken by a really talented PhD student, Anna. And so what she was working on and she was involved in originally was some of the work in sequencing where we identified these CDKN1B mutations in 8% of small intestinal nets. And it's because of that and finding this very low background mutational rate, really started focusing on the methylation, thinking that these tumours could be epigenetically driven. And what Anna did was she took a caseload of over 50 primaries of intestinal nets and looked at sequencing, looked at methylation analysis, and looked at copy number changes. So you can have changes in the way that the genome is made up, which we know can drive cancer development. And what she found with this molecular analysis is just around half of our cases had changes of loss of heterozygosity, LOH, chromosome 18, and they also had specific methylation panels. So this is this top molecular group here. So specific methylation pattern, chromosome 18, LOH. There's a second group here which had very few copy number changes at all. So on the left here, specific methylation pattern. And then this bottom group here had quite a lot of copy number change, and again, specific DNA methylation pattern. And in terms of the CDKN1B mutations, it just fell through this whole cohort. And then what Anna found was the different molecular subgroups had different clinical outcomes. So the 18 LOH group did very well, had the best progression-free survival. This is follow-up of over 10 years. And then the lowest group here with the poorer progression-free survival was the group with the most copy number change. So that was really important and interesting findings, because all of those tumours look the same down the microscope. So in our tumour board, we would have thought they were all the same, but you can see here by the molecular analysis, they've got quite different clinical outcomes. And then another big piece of work, which is still ongoing, and this is with collaborators at Harvard and UCSF, is that we know that small intestinal nets are commonly multifocal. So throughout the small bowel, in around 50% of cases, there's more than one tumour there. And historically, it was thought that these all had arisen from the same clone, so it had a clonal evolution. And we've been applying genomic analysis to really understand the background of where these multifocal intestinal nets arose from. And each of these white arrows here in these two samples cases can show where these different multifocal cases are. So one here, this case had 25 different primaries. This one here had 18, for example. So I'm going to give an example from one patient, 852, where there were 18 primaries, 3 lymph node metastases, and some normal bowel. So this is one of the most common scenarios you might see in terms of small intestinal net pathology. It's in the ileum, it's low grade, it had some lymph node involvement, positive for chromogranin synaptophysin, and the key 67, which I mentioned earlier, was low grade, less than 2%. And it's Eric Nakakura, a surgeon at UCSF. He has been phenomenal in terms of, at operation, he will palpate the whole of the small bowel. He'll resect each of these primaries. He has frozen these. So all of this work is due to his fastidious sample collection over the last 10 years or so. And what we found almost straight away, just by doing sequencing analysis, so just looking at the DNA in the first instance, is that these different primaries weren't clonal. So they were independent clones, which threw over some of the first original thoughts about how these multifocal tumours developed. And we worked this out by looking at the pattern of the mutations, so synonymous and non-synonymous mutations in the different types. We also looked for different types of mutations, so frame shift, what we call missense, what we call deletion. So we can see, you can see from this plot that clearly these different primary tumours were different. So they were independent clones. And what my group have brought to this piece of work is then the methylation analysis of the multifocal small intestinal net cohort. So the cohort that my group's been working on, there's some normal epithelia, there's 79 different primaries across different cases, and then there's 12 metastases. And so what we've been doing by looking at whole genome, reduced representation by sulfite sequencing, so moving away from the array base that I mentioned earlier. So this is looking across those whole 27 million CpGs across the genome, looking at methylation patterns. We've been applying epigenetic clocks that I mentioned to see if the different primaries arose at different times. And we've also been applying methylation patterns to work out the cellular makeup of these different types of tumour. So one of the challenges is when we're looking at data from bulk tumour, is then trying to pull all of that apart in terms of our downstream bioinformatic analysis. So if you think about a bulk tumour sample, you're going to have normal cells alongside tumour cells. So that can dilute the signal from the DNA that might be changed in the tumour cell compared to the normal cell. And then you can also have this different copy number. You can have different numbers of the same chromosomes, you can have different copy number variants. So that can really cause challenges to the interpretation of our data analysis. But there's really, I'll move myself because I've covered her head here. So we've really talented PhD student and now postdoc in the collaborators lab, Peter Van Loo's lab at the Crick, along with Stefan Beck at UCL. This is Lily. This is work from her PhD, and she's developed a brilliant pipeline for analysing this RRBS data in cancer, where you have your bulk tumour sample, you have your adjacent normal sample, you create your RRBS data, you have bulk methylation rate and your normal methylation rate, and then you can determine the purity of the tumour, the copy number of the tumour, and then you can identify and work out the tumour methylation rate. And at that point, you can then start looking at tumour normal comparisons. So we've been applying this to our multifocal tumour cohort in this instance, but we've also have some cases where we've looked at different regions of the same tumour. But the data today is around multifocal tumours. So I will just cover Lily back up and move on. And so this is what we've seen as our first shot over the bow. This is unpublished data at the moment. So this is a subsection of our cohort, and we're looking at the 5,000 most variable methylation sites across the genome. But you can see quite clearly that the normal pattern here, the normal tissue here, is very, very different methylation patterns. The yellow being high degree of methylation, the blue being low degree of methylation across the different tumours. But you could also see here, this is all from the same patient. So although these are, you can see these are slightly different, this individual's tumours are broadly clustering together. So this piece of work is ongoing and it's unpublished, but it's just showing you some of the genome-wide approaches and information that we can take away from this type of analysis. We can also identify differentially methylated regions between the tumour and the associated normal. And so far across the genome, we've identified nearly 4,000 of these differentially methylated regions. We're picking out and identifying some interesting genes which might be implicated in tumour development. And we can also look at how the methylation level changes as you just walk across the length of the chromosome in a different site. So we get some really nice visualization of where the methylation occurs across the genome. And something that's really important is we know that methylation changes outside of the coding region, so outside of the exome and the bit that's red for gene expression can also have a big impact in cancer development. And I'm going to go on and tell you a little bit about our application of epigenetic clocks. I'll move myself again. So this is work by Amy Webster, who's a really talented postdoc in my group. And she's been applying different types of epigenetic clocks to see if we can learn about how and when these different multifocal primaries arose. So we know that these clocks use certain markers of DNA across the genome to give us an estimation of chronological aging. We can use them to give us an idea of biological aging, and we can also use different clocks for mitotic clocks, which gives us an approximate idea of the number of cell divisions. We might imagine and expect that cancers have more and a higher number of cell divisions, or might show more sort of biological aging or move forward more quickly. So Amy looked at and applied all of these clocks, but I'm just going to give an example of one of the clocks that she used. So this is across all of the samples in our cohort. So this is the age and the age-adjusted acceleration using one of the epigenetic clocks in the normal epithelia. And this is the primary tumour, and then you can see there's that shift in the age and the metastasis. So we think that the sort of the acceleration, the age acceleration is higher between the normal and the tumour, and then further again between the tumour and the metastasis. And this shows data across all of our different cases. So what we have here is this is all the multifocal primaries from one case. This dot here, the orangey dot, is the actual age of that individual. Then the greeny dot is the the epigenetic clock, the estimated age of the normal tissue, and then these are all the cancers. So there's age acceleration in different tumours, but you can see the different individual tumours have a different age. And it's the first time that this approach has been demonstrated and been used in neuroendocrine tumours. And it also really highlights that these different tumours are arising at different times, and they are different. They're independent clones and have their own clonal development and evolution. I'm now going to move on and talk about some of the work we've been doing in pancreatic neuroendocrine tumours. And this is work mainly by Teresa, Chris, and Alison. So again, when the initial efforts and work around looking at the DNA sequence of pancreatic NETs was undertaken over 10 years ago, it's 12 years ago now, in fact, one of the biggest and most important findings is around 40% of these tumours harbour mutations in HRX and DAX, which are these chromatin remodelers. They're an important part of how our DNA folds up and is packaged into the nucleus. And Oral Perrin's group in Bern published really important findings from this, following on from this, is that if tumours had mutations or had lost expression of HRX and DAX, they had a worse clinical outcome. So this is showing progression-free survival and overall survival across the different groups, so either if they'd lost HRX or DAX, or, and it's a phenomenon that's associated with HRX or DAX loss, if the tumours demonstrated this alternative lengthening in telomeres. So we have finally found a marker, a DNA-specific marker, that gives us some insight into the prognosis. So we know that tumours that harbour HRX and DAX have a worse outcome. That's something for us to bear in mind clinically. So my group took this information and we thought long and hard about how DAX and HRX interacted. We could see that HRX interacts with de novo, so new methylation across the genome. DAX interacted with DNMT1, which is involved in maintenance DNA methylation. So what we hypothesized is that if there are mutations in DAX, you might expect to see more methylation changes across the genome because it's involved in maintenance rather than de novo, new methylation. And that was exactly what we saw. And moreover, we identified that if patients had a mutation in just HRX or DAX, they had a different outcome. So this right-hand figure here, the top line is if the tumours didn't have mutations in HRX or DAX. The blue line is if they'd lost DAX, and then the green line is if they'd lost, the blue line is if they'd lost HRX, and the green line is if they'd lost DAX. So tumours that had harboured mutations and lost expression of DAX had the worst outcome. So that gave us a little more information around what this molecular analysis and profiling could do. We then went on, move myself again, have an ongoing collaboration with Oral Perrin's group in Bern, and then we started looking at integrating the mutational status with methylation analysis. So this was all again on an array-based approach rather than the sequencing-based approach that I was talking about in the small intestinal nets. This is quite a complicated figure, but I'll talk you through it. So what we did with this cohort of pancreatic endocrine tumours, we looked to see if they had mutations in MEN or DAX or HRX, and they fell into three groups. So this left-hand group harbour mutations in MEN, and also harbour mutations in DAX and HRX. And when a group harboured mutations in MEN but hadn't lost HRX or DAX, and this last group didn't have mutations in MEN1 or HRX and DAX. And you can see there are three very distinct groups with different methylation patterns. So we could see that there is this relationship between the underlying mutations and the methylation pattern. We've then gone on to apply methylation analysis to see if we can actually identify cell of origin in pancreatic nets. So it's widely accepted that pancreatic nets arise from islets of Langerhans. And there's also increasing evidence that non-functioning tumours arise from alpha and beta cells, but the cell of origin remains unclear. And the mechanisms of how these tumours progress is, again, really unclear and largely unknown. So looking at an islet, you have the alpha and beta cells, but you also have delta cells, you have pancreatic polypeptide cells, exocrine cells. So you have different cells that make up the islet. And what we did collaboratively is we looked at a cohort of 125 pancreatic NET cases, and with that we looked at methylation, so the 450K methylation array. We looked at the sequencing, so there's whole genome sequencing data from 80, and we finished off the cohort by doing targeted sequencing of our genes of interest on the rest of the 45 cases in the cohort. And we also fed in as a reference methylation profiling of sorted alpha and beta cells. So we used that as our reference to see if these tumours were most identified and might have had a cell of origin that related to alpha and beta cells. And when we applied this approach, we could see that this group up here were alpha-like. So here's the alpha cells that underwent the methylation profiling. This column here shows whether there are HRX or DAX mutations, of which there are very few. MEN1, quite common, and very few copy number changes. So these alpha cells had MEN1 mutations, very few copy number changes. These were alpha-like tumours. The bottom we have beta-like tumours. Here's the beta, had very few HRX or DAX mutations, very few MEN1 mutations. But there's all of these tumours in the middle which don't really fall into these neats. So we don't really know if it's related to delta cells, or other cells in the islet, or a combination of the above. And then when we looked at the progression-free survival and outcome for those patients, the alpha-like tumours did less well than the beta tumours. So you have the intermediate tumours who did the worst, so that big group in the middle, the alpha-like tumours here, and the beta-like tumours did the best. And if you remember, the beta-like tumours didn't harbour mutations in HRX or DAX or MEN1. So that was really insightful, but it just gave us a glimpse to start putting together a picture of how these tumours might develop. So what we can see and what we think so far is you have these alpha cells, where you may have MEN1 mutations, which then lead to an alpha-like tumour. What might happen next is HRX or DAX mutations to then push on and drive on to a different type of tumour with mixed or loss of differentiation. Of beta cells where we haven't identified mutations in HRX or DAX or menin, so a beta-like tumour, which I've just shown has a better outlook in progression-free survival. Then we have all of these other tumours which may come from other progenitor-like cells, might harbour HRX and DAX mutations. But with this approach, we're beginning to piece the jigsaw together. And then the last area I'm going to touch on today is our most recent work, which is focused on pancreatic nets again, but it's actually creating a methylation atlas over a number of tissues, which we can then apply to pancreatic nets, where it includes all of those different cellular types in the islet. So this was published last year, was led by a colleague, Andrew Teschendorf. We contributed to the work in the pancreas and the pancreatic nets. But what he's created is a DNA methylation atlas where you can identify and infer a methylation profile and create a matrix based on single cell RNA sequencing data. So that's really powerful and a useful approach for rarer and difficult to access tissues, such as the pancreas, such as the brain. So what this piece of work did, it focused on 12 different tissue types from brain, lung, heart, liver, and pancreas, etc. Used single cell sequencing. It was then matched and ran matched bulk expression data and DNA methylation data sets were run alongside the single cell sequencing, where it was then imputed to a DNA methylation matrix. And then that was your normal tissue matrix based on methylation for that tissue type. And it was kind of based on hardwired methylation changes at promoter regions. And it was usually between 1 and 300 genes we used as that hardwired matrix, which was then used against our different tumor types. So this was just showing some validation from this single cell RNA sequencing data across and validating across different tissue types with bulk tissue. So here's the pancreatic site here, the different cellular types, so epithelial, endocrine, immune, fibroblast, and neuroectoderm. There's no neuroectoderm in the pancreas, but these are the different cellular types that were picked up in the matrix. So this is a single cell RNA-seq data, and these different cellular types were then identified and a matrix was built for pancreatic tissue. So bearing in mind with the work that I showed you with colleagues in Bern, we just had data from alpha and beta cells which were fed in. So the reference was fed in. In this situation, the reference is created, but we have a reference for all of the different cells that make up the pancreatic islet. And then we used data that my group had produced from 57 pancreatic nets to then look at the different cellular makeup. And this is in comparison with pancreatic adenocarcinomas taken from the Cancer Genome Atlas. And you can see, just sort of having a high level look across, you can see there's differences in the proportion of acinar cells, clearly in alpha and beta cells, as you might expect for pancreatic nets, but also in the immune cell makeup between pancreatic and adenocarcinomas. And also when we did apply the matrix to the Cancer Genome Atlas pancreatic adenocarcinomas, we identified a handful of cases which had been misclassified as adenocarcinoma. They were in fact pancreatic nets, which then when we went back into the Cancer Genome Atlas, our matrix had more accurately identified that tumour type. So by using this approach, we're now, for the first time, really beginning to understand the individual cellular makeup of our different tumours. I think it's a really, really exciting area and a really exciting advance to be involved with. So in summary, I think over the last, it's been a bit of a tour de force, but over the last 25 minutes or so, I think you'll see, and I hope you'll appreciate, that with all of our genomic sequencing and analysis in neuroendocrine tumours over the last 10 years, we can see they've got a very low background mutation rate. Unfortunately, there are very few actionable mutations that we can use some of the more commonly used molecularly targeted therapies in cancer against. Pancreatic and bronchial nets quite often, in around 40% of cases, have mutations in the epigenetic machinery. But if we integrate methylation and expression and DNA sequencing analysis, we can identify molecularly distinct groups in intestinal and pancreatic tumours, which are tied in with progression-free survival and outcome. We can use epigenetic clocks to determine the origins of multifocal tumours. We see those different timings of the different multifocal tumours. And finally, we've been doing some work looking at the methylation analysis to identify the cell of origin and cellular composition of neuroendocrine tumours. So that's just a wrap up of some of the things we've been doing over the last 10 years or so. And with that, I'd just really like to thank all of my national and international collaborators, all of the really talented PhD students and postdocs that have worked with me and with my group over this time. But most importantly, I'd like to thank all of our patients, because without their engagement and their encouragement, we would have no tissue to work on and no data to present today. So with that, I'd just like to thank you again for the very kind invitation, and I'm sorry I can't be joining you in person today, and I hope you enjoy the rest of the meeting. Thank you.
All right, so that concludes our talk by our keynote speaker, Dr. Chrissie Thirwell. So at this time, we're going to move on to our next speaker that I'm going to introduce. You'll just give me a moment to pull up his talk. All right, this looks good. Okay, so I'm going to go ahead and move forward and introduce our next speaker. Our next speaker is Dr. Ajaykumar Morani. He is an associate professor of abdominal radiology and associate director of quantitative imaging analysis core at the University of Texas MD Anderson Cancer Center. He is also the co-founder and co-chair of disease-focused panel on abdominopelvic neuroendocrine neoplasms of the Society of Abdominal Radiology. He has co-directed multiple hands-on workshops and webinars, and also co-authored several publications in the field of imaging abdominopelvic neuroendocrine neoplasms. And his talk today is titled Anatomic and Functional Imaging, including Radiomics for Pancreatic Neuroendocrine Neoplasms, 2023 updates. And here we go.
Greetings. My presentation is on anatomic and functional imaging, including radiomics for pancreatic neuroendocrine neoplasms. I do not have any relevant disclosures. So pancreatic neuroendocrine neoplasms are a heterogeneous group of neoplasms with neuroendocrine differentiation. They are also called as islet cell tumors. They account for about 7% of all pancreatic cancers. However, their incidence is increasing with more and more usage of cross-sectional imaging and endoscopic ultrasound. Up to about 10% of the pancreatic tumors in autopsy studies have been found to be neuroendocrine neoplasms. In general, gastroenteropancreatic neuroendocrine neoplasms are very diverse clinically and biologically. Their treatment and prognosis depend not only on staging and primary site, but it also depends on histological grading. Grade 3 poorly differentiated neuroendocrine cancers are aggressive and nearly always advanced at diagnosis with poor prognosis of about 10 months survival. Grade 1 and grade 2 well-differentiated neuroendocrine tumors, they can be indolent with median survival of 8 to 16 years. And now we have a new category of grade 3 well-differentiated neuroendocrine tumors with intermediate prognosis. If we see different neuroendocrine neoplasms by different disease sites, the pancreatic neuroendocrine neoplasms have worse prognosis overall with less median overall survival, and the median overall survival also decreases with increasing grade of these neuroendocrine neoplasms at any disease site, including the pancreatic site. From genetic point of view, neuroendocrine tumors tend to harbor MEN1, DAX, and ATRX gene alterations, while neuroendocrine cancers tend to harbor P53 and RB gene alterations. So taking into account all these factors of grading, differentiation, and genetic alterations, WHO recently updated its classification of pancreatic neuroendocrine neoplasm. Now, basically in neuroendocrine tumors category, we have grade 1, low grade, grade 2, intermediate grade, and high grade neuroendocrine neoplasms. All these three grades are well differentiated. When they are well differentiated, they are called as neuroendocrine tumors. When they are poorly differentiated and high grade, they are called as neuroendocrine carcinomas, which are small cell and large cell neuroendocrine carcinoma. The basic grading is still based on Ki-67 index and mitotic count, less than 3%, 3 to 20%, and then greater than 20% for grade 1, grade 2, grade 3 neuroendocrine tumors and carcinomas, and mitotic count less than 2 to 20, greater than 20 count, respectively for grade 1, grade 2, grade 3, and the high-grade neuroendocrine carcinomas. So for Ki-67 index and mitotic count, we need pathology for this of the specimen in these patients. For pathology, so we need to do biopsy or excision. When we do biopsy, usually it's a single, most of the time it is a single location biopsy. Maybe we can do few locations, but we cannot biopsy all the lesions or many sites in a particular lesion. Then a single location biopsy may not be representative of the whole tumor heterogeneity within a lesion or from one lesion to another lesion in a patient with multiple disease sites. And also we know the grade transformation can occur, so the aggressiveness of the disease may increase over time, and so the single time point biopsy may not be representative of the future aggressiveness of the tumor. Imaging in this sense can help us to direct tissue sampling from more aggressive disease sites. Not only that, in a non-invasive way, if we can assess the qualitative and quantitative imaging features, including radiomics, we may get an entire picture of a whole lesion or from one lesion to another lesion, and it may serve as a potential virtual biopsy in a patient. Regarding HSCC staging system, we know that still recently pancreatic neuroendocrine neoplasms were staged just like pancreatic ductal adenocarcinoma, but now it has changed. The poorly differentiated pancreatic neuroendocrine neoplasms are staged just like pancreatic ductal adenocarcinoma, but the well-differentiated neuroendocrine neoplasms, now they have different staging on their own. So less than 2 centimeter, 2 to 4 centimeter are T1 and T2 tumors respectively. Greater than 4 centimeter size or invading duodenum or common bile duct are T3, while those invading the adjacent structures are T4 tumors. Pancreatic neuroendocrine neoplasms can be hereditary or sporadic. Up to about 20% of pancreatic neuroendocrine neoplasms are associated with some hereditary cancer predisposition syndrome. Most common of them are MEN1 and VHL. MEN1 pancreatic neuroendocrine neoplasms are usually larger. They are likely benign and up to 15% are cystic. Mostly they are non-functioning tumors, and multifocal gastrinomas are more commonly found in these cases. In VHL, pancreatic neuroendocrine neoplasms tend to be multiple, usually small, less than 2 centimeters in size, and up to 15% are malignant in this patient. Most of them are non-functioning. Here are a few examples. We have a pancreatic tail mass in this patient, avidly enhancing, suspicious for neuroendocrine neoplasm. Patient also has multiple solid renal masses, suspicious for renal cancers. And also patient had a right adrenal pheochromocytoma, multiple renal cysts. Putting all this together, this is a case of hereditary pancreatic neuroendocrine neoplasm in the setting of von Hippel-Lindau syndrome. Another patient, 36-year-old man with recurrent seizures. Patient had multiple subependymal calcific tumors along the ventricular walls on CT scan with the brain and enhancing pancreatic tail mass. So it's a case of hereditary pancreatic neoplasm. This was in selenoma in this patient with tuberous sclerosis. Pancreatic neuroendocrine neoplasms can also be looked in a way of functioning and non-functioning. Non-functioning tumors account for the majority, so the larger category of the pancreatic neuroendocrine neoplasms, while functioning tumors account for 30% of these neoplasms. Functioning tumors, because they produce hormones, they cause symptoms and syndromes, and so they are detected when they are small, and they are usually symptomatic. And these are the kinds: insulinoma, gastrinoma, glucagonoma, serotonin-producing tumors, ACTH hormone-producing tumors, etc. But again, the big chunk is non-functioning. About 50 to 70% of pancreatic neuroendocrine neoplasms are non-functioning ones. And most common pancreatic neuroendocrine neoplasms, even in that hereditary predisposition syndromes, are non-functioning ones. Although they are non-functioning, they still secrete hormones, but these are without symptoms or syndromes. Most of them are sporadic and solitary, but when they are seen with familial predisposition syndromes, they tend to be multiple. As these are non-functioning, they tend to grow larger, and when they are detected, they are larger in size. And they are often heterogeneous due to necrosis or cystic changes. About 1/3 of them may have tumor thrombus in the adjacent vein, and 60 to 80% metastasize, had already metastasized at the time of diagnosis, often to the liver and nodes. If we look into functioning pancreatic neuroendocrine neoplasms, most common are insulinomas and gastrinomas. Insulinomas are the most common of all the functioning pancreatic neuroendocrine neoplasm. They tend to be solitary, 10 to 30% associated with MEN1, where they can be multiple. Most of them are usually benign, grade 1, less than 2 centimeters in size, and often cured by surgery. When they are malignant, they are often grade 2 and diagnosed due to local invasion, nodal or hepatic metastasis. The size is very important in this. As you see, less than 2 centimeter size insulinomas have about 100% 10-year survival, compared to only 30% when they are more than 2 centimeters in size. Gastrinomas are the second most common functioning pancreatic neuroendocrine neoplasms, often multicentric. 80 to 90% occur in gastrinoma triangle. 60 to 90% of them are malignant, and 1/3 associated with liver and bone mets. Regarding imaging of pancreatic neuroendocrine neoplasms, we have anatomic imaging and functional imaging. In anatomic, basically ultrasound, endoscopic ultrasound, CT, MRI. Just few brief points about endoscopic ultrasound here. Later on, we'll just switch to CT and MRI and PET from functional imaging point of view. So endoscopic ultrasound plays a crucial role when pancreatic neuroendocrine neoplasm is suspected, and especially this is true for the small tumors less than 2 centimeters in size, like insulinomas, gastrinomas. And it is the study of choice, especially when CT and MRI fail to detect the pancreatic neuroendocrine neoplasm. And the another very important use is we can do fine needle aspiration at the same time of endoscopic ultrasound. From functional imaging point, it's all PET-based imaging now. And then radiomics can also be very useful, as we will see. And this is the field being developed in this setting. Regarding functional imaging, octreoscan was the most popular and was the first to be used from the functional imaging point of view in these cases, with very high specificity of 92 to 100%, but lower sensitivity 40 to 100%, but still better for well-differentiated and larger lesions. Now, PET-CT is of choice due to its superior spatial resolution, and it plays actually a crucial role when CT and MRI are negative. Gallium-labeled somatostatin analogs were recently approved by FDA, replacing Octreoscan, especially DOTATOC, gallium dotatate, DOTATOC, and DOTANOC. PET-CT is now preferred for lower grade neuroendocrine neoplasms with very high sensitivity and specificity varying from 88 to 95%, 94% accuracy for insulinoma, and so very important preoperative role in these cases. But we have to be careful because of the physiologic uptake of this agent in incinate process and adrenals when we are evaluating the disease sites, especially in the uncinate and adrenals. FDG PET-CT can be very useful for aggressive neuroendocrine neoplasms, the higher grade ones. So as we see in this patient, we see multiple hypermetabolic nodules in the pancreas and also in the duodenum, multiple hypermetabolic nodules. This patient had suspected neuroendocrine neoplasm. These were multiple gastrinomas, and these were not seen when the CT scan was done or even on MRI. From anatomic imaging point of view, at least one high quality CT or MRI is useful for localization staging. Functioning lesions usually show typical hypervascularity with slow washout. CT is useful and has a 69 to 94% detection rate, but it failed to detect about 68% of lesions less than 1 centimeter and 15% lesions 1 to 2 centimeter in size. Similarly, MRI has more value, especially for tumors greater than 2.5 centimeter in size, and these tumors tend to be T2 hyperintense. Diffusion-weighted imaging had significant value in these patients, and it can be comparable to even gallium dotatate PET-CT in detecting the small pancreatic neuroendocrine neoplasms. MRI in particular also plays a very important role in surveillance and screening of these patients. Okay, so standard. From the anatomic imaging point of view, CT of the neck, chest, abdomen, pelvis is recommended as the basic imaging for diagnosing, staging, assessing treatment response, and restaging of primary pancreatic neuroendocrine neoplasms. MRI is preferred for targeted assessments of liver, pancreas, bones, and brain. Somatostatin receptor imaging should also be part of the workup, and especially for lower grade neuroendocrine neoplasms, and FDG PET-CT preferred for the higher grade neuroendocrine neoplasms or neuroendocrine cancers. Okay, let's see the role of imaging from different point of view, like diagnosis, grading, prognostication of these cases, and also for treatment planning. From qualitative imaging features point of view, and typical diagnostic features are avid arterial enhancement and delayed post-contrast washout. 1/3 of these patients can show tumor thrombus, as we see in these patients in the tumor thrombus in the splenic vein, and many of them can present with liver or nodal or bony mets, as we see here, liver metastasis with this large pancreatic tail mass. And the other atypical features can be ductal dilatation, as we see here, distal pancreatic ductal dilatation and some atrophy. Sometimes they may show even intraductal growth. So as we see, although the typical feature is arterial enhancement and delayed post-contrast washout, some of the features may overlap with the features of pancreatic ductal adenocarcinoma. And as we know, pancreatic ductal adenocarcinoma is the more common malignancy in the pancreas compared to neuroendocrine neoplasm. So how to differentiate them? When we see irregular margin of the mass, and particularly if the mass is hypovascular compared to the pancreatic parenchyma and showing delayed post-contrast enhancement, then we tend to favor it as a pancreatic ductal adenocarcinoma. On the contrary, if we see the very well-defined margin of the mass looking more homogeneous in appearance, then we favor it to be a pancreatic neuroendocrine neoplasm. But in real life, there are more challenges. As we see in this patient, we have 3 cases here of pancreatic masses. This one, hypovascular, but showing pancreatic ductal dilatation, was suspected of pancreatic ductal cancer. This is another irregular mass encasing the celiac artery and splenic artery. Was also suspected of pancreatic ductal adenocarcinoma, and this hypovascular mass suspected of pancreatic ductal adenocarcinoma. But as we see, the first 2 are pancreatic neuroendocrine neoplasms. So up to about 57% of non-functioning pancreatic neuroendocrine neoplasms or pancreatic neuroendocrine cancers show overlapping imaging features with ductal adenocarcinoma. In these cases, more qualitative features may help, and also another more quantitative imaging features may help. Like we can use radiomics. What we do in radiomics, basically, we do the image segmentation in this region of the tumor, then we assess the features which are basically not seen by our naked eyes, and the computer extracts different features of these masses like the shape, margin, texture, another voxel heterogeneity, microenvironmental heterogeneity within the mass, and then we try to create some radiomic signature. Once the radiomic signature is created in a model, then this model is validated further to create final radiomic model. The details of this are beyond the scope of the talk, but we know that several radiomic features have been useful in pancreatic neuroendocrine neoplasms. The most common studied ones are entropy, kurtosis, uniformity, skewness, low percentile, and higher percentile. Uniformity is basically, as the name suggests, is the texture homogeneity within the tumor, right? Entropy, kurtosis, skewness are the heterogeneity. When we try to assess the heterogeneity of the attenuation on CT or intensity on MRI at the level of the whole tumor, at the level of the whole image, then it is entropy. If we try to assess the same heterogeneity at the level of voxel, it is called as kurtosis. If we go even more deeper at the microenvironmental heterogeneity, then it is called as skewness. Lower percentiles often represent cystic degeneration and necrosis within the tumor, while the higher percentiles reflect the blood perfusion. So radiomics can be useful for diagnosing pancreatic neuroendocrine neoplasm. I mean, as we know that the functioning neuroendocrine neoplasm is not a problem because they produce symptom syndrome. So we know, okay, this one is a neuroendocrine neoplasm. When they show typical features, arterial enhancement, washout, then again, not a problem. These are those atypical ones or the non-functional pancreatic neuroendocrine tumors which pose diagnostic challenges and difficult to differentiate sometimes from pancreatic ductal adenocarcinoma. In such cases, when we see higher uniformity, homogeneity greater than 0.34 or lower entropy, heterogeneity less than 1.89, that suggests it is more likely a pancreatic neuroendocrine neoplasm rather than ductal adenocarcinoma. Lower skewness, that is lower heterogeneity at the macroenvironmental level, lower heterogeneity suggests neuroendocrine versus ductal adenocarcinoma. This is an example of pancreatic tail mass showing less kurtosis and less skewness in this patient, favoring it as a pancreatic neuroendocrine tumor. Pancreatic head mass showing higher kurtosis, higher skewness. Higher kurtosis, that means higher heterogeneity at voxel level, skewness at the macroenvironmental level. So higher heterogeneity within the mass suggests it's a pancreatic ductal adenocarcinoma. So radiomic actually worked very well in this patient. Radiomics can be even that much useful that it may outperform the clinical or radiological features for atypical non-functional neuroendocrine tumors, as shown in some studies. If we use radiomics and combine it with clinical and radiologic features, it may even prove even more robust model to differentiate neuroendocrine neoplasms from other solid pancreatic masses or ductal adenocarcinomas. Just like CT on MRI also, we can do a heterogeneity assessment on ADC histogram. We can do texture analysis, and some studies have shown that this can be very useful in these patients. Okay, let's go to the grading part now. Can we differentiate the grading of the pancreatic neuroendocrine neoplasms? As we saw, grade 1 to grade 3 have different prognosis, and so the management can also differ. As we see here, 3 cases. All these 3 patients are cases of pancreatic neuroendocrine tumor. The first one, cystic mass with rim enhancement. Second one, a solid mass with central calcification. Third, another solid mass with well-defined margin, but little bit less ill-defined, like ill-defined interface with the pancreatic parenchyma. We see first is low grade, intermediate grade, second one, and the higher grade, the lower one. So although these are all neuroendocrine neoplasms, they have different appearances. And so the different grades of neuroendocrine neoplasms can have different appearances. And if we can diagnose that, we can assess the grading of these tumors even before doing biopsy or without needing biopsy. So there are some features which have been found useful in grading these neuroendocrine neoplasms. If we know that, okay, this patient has a pancreatic neuroendocrine tumor, now I have to assess the grade. If I see the well-defined margin of the mass, if I see T2 hyperintensity of the mass, those features suggest that it's a lower grade neuroendocrine tumor. On the other hand, if we see irregular shape of the mass, ill-defined, encasing the vessel like aggressive looking tumor, and also it has T2 dark signal, that suggests that this is a higher grade neuroendocrine neoplasm. If the pancreatic neuroendocrine tumor showed avid arterial enhancement, then we can suspect that this is probably a grade 1 or grade 2 neuroendocrine tumor, because the lower grade tumors have higher microvascular density. Not only these qualitative features, we can assess quantitatively, like tumors greater than 2 centimeter tend to be more higher grade, grade 2 or higher grade. We can also assess the ratio of the tumor attenuation or signal intensity versus the background pancreas on CT or T2 weighted MRI. On CT, if it is more than 1.1, it suggests grade 1 tumor. On MRI, greater than 1.8 on T2 weighted imaging suggests lower grade tumors. ADC values can be also useful. Lower ADC values are generally found in the aggressive ones. So higher grade neuroendocrine neoplasms or neuroendocrine cancers will have lower ADC values, especially less than 1.19 for grade 3 tumors. If we go even lower, less than 0.69 found in poorly differentiated cancers. Here is a study with a few images, grade 2 neuroendocrine tumor. On the left side, we can see T2, relatively T2 bright mass, and then showing restricted diffusion. Here is grade 3 neuroendocrine neoplasm with a T2 dark signal, hypovascular mass, restricted diffusion. In this study, they found that 0.95 ADC value cutoff was better in differentiating neuroendocrine cancers from grade 1 and grade 2 lower grade tumors. Okay, so far we saw how we can differentiate grading on CT and MRI. We can use PET-CT. PET-CT actually has been found even more useful, and it is having increasing role in these patients. Like in this patient, we can see avidly enhancing pancreatic mass, intensely showing intense uptake on the gallium-dotted PET-CT. So when they show intense uptake on gallium-dotted PET-CT, that means they have a lot of somatostatin tracer receptors. And in this case, this mass was not much avid. It was actually showing minimal avidity on FDG PET. So if we see intense avidity on gallium-dotted PET-CT, that suggests it's a lower grade neuroendocrine neoplasm. Here is another patient, intensely avid pancreatic head mass, showing intense uptake on gallium-dotted PET-CT. So again, intense uptake on gallium-dotted PET-CT suggests lower grade or well-differentiated neuroendocrine tumor. Well-differentiated will have a lot of somatostatin receptors. On the contrary, here is another patient with pancreatic mass here and multiple liver mets. And if you see, liver mets are photopenic, actually not showing activity on gallium-dotted PET. But on the contrast, they show intense uptake with the FDG PET. So if we see intense uptake with FDG PET, they tend to be higher grade neuroendocrine neoplasms or neuroendocrine cancers. And FDG avidity actually has been found in increasing if the Ki-67 rises more than 15%. So if FDG avidity increases, that suggests higher grade neuroendocrine neoplasm or neuroendocrine cancers. So overall, we saw if the pancreatic mass has well-defined margin, if they are T2 hyperintense, if they show avid arterial phase enhancement, and if they show intense uptake on gallium-dotted PET, they tend to be lower grade neuroendocrine neoplasms. But if they have ill-defined intersurface with the pancreas, if they have irregular shape, encasing vessels, T2 dark signal, and if they show intense uptake on FDG PET-CT, then they tend to be higher grade neuroendocrine neoplasms or neuroendocrine cancers. Similarly, radiomics can also be useful because sometimes even after studying these imaging features, we may not be able to differentiate the grade. So radiomics has also been found useful. Entropy, the more sensitive single factor in helping to differentiate the grades of the neuroendocrine tumors. Skewness and kurtosis, as we saw, these are heterogeneity markers. These values have been found to increase as the tumor grade increase, logically speaking. But in real life, sometimes radiomics may not work. It has been found in several studies to work in majority of the patients, but in several cases, it may not work. As in this patient, we can see intensely avid-enhancing mass here, neuroendocrine tumor with higher kurtosis, although it is a low-grade neuroendocrine tumor. And this is a higher-grade neuroendocrine tumor, but it has a lower kurtosis. Although skewness and entropy are trending with low-grade and high-grade neuroendocrine tumors. So radiomics may not work all the time, and there is a lot of work which is needed in this field. So far we saw diagnosis and grading point of view how imaging helps. So imaging can also help from the prognostic point of view. If we see pancreatic ductal dilatation with pancreatic neuroendocrine tumor or liver mets, tumor size greater than 3 centimeters, these are strong predictor of poor prognosis. Irregular lobulated contour, bigger size, necrosis, vascular invasion, these are all poor prognostic factors. SUV values on dotted PET-CT have also been found useful, but we have to be careful because technical factors may change the SUV values too. Overall, it has been found that those with SUV max greater than 13 on gallium dotted PET-CT were found to have better prognosis, better progression-free survival, and predicted also response to the Lutathera therapy, PRRT therapy, which was recently approved. Imaging can also be useful for treatment planning. Basically, pancreatic neuroendocrine tumors less than 2 centimeters in size can be managed with just surveillance or follow-up every 6 months for 2 years with cross-sectional imaging and then yearly for temporal evaluation. If the non-functioning neuroendocrine tumor is greater than 2 centimeters in size or any size functioning localized neuroendocrine tumor is there, then they can be managed with resection. If the patient has multiple tumors along the pancreas or affecting major portion of the gland, then they may need total pancreaticoduodenectomy. When these are localized grade 1 or grade 2 neuroendocrine tumors, they are usually resected without additional treatment needed after that. But when they are grade 3 tumors or higher, then they need additional radiation or chemotherapy. Surgery may be performed sometimes as a palliative reason for advanced neuroendocrine cancer. Those patients with liver mets can also be treated with liver-directed therapy, and it has been found to have approximately 50% response rate. Even surgery can be performed even though patient may have liver mets. When it can be performed, if the patient is fit, surgery is feasible. If the patient has metastatic disease, which is more superficial, more localized in any particular lobe or segments, or if the patient has oligometastatic disease. And sometimes even combination of therapies with surgery, ablation, radiation can be performed. Here we have an example of an 18-year-old girl with von Hippel-Lindau syndrome, a partially calcified pancreatic neck mass. We can see here the patient also had only single left hepatic lobe metastasis. So disease is confined basically to the left lobe, liver, and pancreas. So patient underwent Whipple surgery and left hepatectomy. Another patient who had prior distal pancreatectomy found to have these 2 liver mets, and these were in different segments. Although wedge resection was contemplated, but going towards less invasive type of treatment, ablation was planned. And the anterior ablated lesion showed good post-ablation changes without recurrence, but the posterior ablated lesion after some time started showing recurrence along the margin, needing second ablation again. So not only surgery, imaging can also be useful to plan and triage systemic therapy in these patients. Systemic therapy is usually needed for locally advanced pancreatic neuroendocrine neoplasms or those with distant metastatic tumors. And PET-CT has found an increasingly important role in these cases. Recently, PRRT therapy, Lutathera therapy was approved by FDA, and it is now being widely used for progressive somatostatin receptor positive gastroenteropancreatic neuroendocrine tumors. So to triage these patients, actually PET-CT has been found very useful, and some people even recommend doing dual tracer PET-CT with somatostatin tracer agent and also PET-CT with FDG. So the tumors that bind to the somatostatin receptor agents like gallium dotatate but are not FDG avid, they tend to be the lower grade, as we saw well-differentiated neuroendocrine tumors, and then they respond where they need treatment with somatostatin analog or PRRT therapy. On the contrary, those pancreatic neuroendocrine neoplasms which show intense FDG avidity and less or no avidity on dotatate, they tend to be poorly differentiated. So When we see intense FDG avidity and less or no avidity on DOTA, then they need some kind of platinum-based chemotherapy. But in real life, 35 to 50% will show some overlap, DOTA avidity, FDG avidity both, and they may need different therapies, somatostatin analog, PRRT, CAPTEM, or combination therapies. So PET-CT can be useful to triage these patients in different kinds of systemic therapies. Here we have an example of a pancreatic mass with multiple liver mets, intensely hypermetabolic on DOTA-TET, gallium DOTA-TET PET-CT, because these are intensely avid on gallium DOTA-TET PET, that suggests well-differentiated or lower-grade neuroendocrine tumor. It also predicts good prognosis. It also predicts good response to the Lutathera therapy, PRRT therapy. So that was given in this patient. Pancreatic mass significantly shrank down in size. Liver mets also significantly improved. So patients showed significant positive post-treatment response with Lutathera therapy in this patient. Another patient with intensely hypermetabolic pancreatic mass with multiple liver mets with intense activity on FDG PET, because these showed intense uptake on FDG, favor poorly differentiated cancer or higher grade neuroendocrine neoplasm, and so needing platinum-based therapy. This patient was treated with platinum-based therapy. Pancreatic mass almost nearly resolved. Liver mets significantly improved, but there was this residual one needed for the treatment. But overall, PET-CT helping to triage these patients for systemic therapy. As we saw earlier, in real life, there will be many patients who will have disease. Some disease avid on DOTA-TET PET, some disease avid on FDG PET, some disease avid on both. And these patients will need this middle line, multiple lines of therapies. So gastroenteropancreatic neuroendocrine neoplasms are very diverse clinically as well as biologically, ranging from indolent neoplasms to very aggressive neoplasms. Their treatment and prognosis depend not only on the stage as well as grade, differentiation, site, and several other comorbidity factors. Traditional imaging evaluation for diagnosis and staging does not yet yield sufficient information. So we need more qualitative features, quantitative imaging features to be assessed, including functional imaging with diffusion-weighted MRI, DOTA-TET PET-CT, texture analysis with radiomics and radiogenomic features, which all appear promising for more accurate staging, tumor risk stratification, guiding management, and assessing treatment response in these patients. Thank you so much for your kind attention.
All right, so that concludes our second talk from Dr. Morani. And there was a chance he was going to be here for questions depending on his schedule, but I don't currently see him. So we have about 5 minutes before our next set of this conference, which we're really excited about. Our next section will feature talks from 2 trainees in the field of neuroendocrine cancer research. So I'm okay to go ahead and proceed. Angaraj, are you okay to introduce our next speakers?
Yes. Right. Thanks, Rachel. So the next talk is from Dane Tow. The title is Targeting Serotonin Metabolism Neuroendocrine Cancers. Dane is currently a postbachelorate research trainee in the lab of Dr. Poe at the University of Iowa. He studies small bowel neuroendocrine tumors with a focus on serotonin metabolism. He plans to continue his research training in a cancer biology graduate program. Without further ado, I'll welcome Dan to give this talk.
Thank you for the introduction and the opportunity to present my research today. So I'm just going to share my slides here. So as you mentioned, I'm from the EAR lab at the University of Iowa, and I will be talking about serotonin metabolism in neuroendocrine tumors. So our lab studies small bowel neuroendocrine tumors, which are one of the most common subtypes of NETs, accounting for just over 50% of this type of tumor. And one of the characteristics that we're interested in is that these tumors tend to overexpress the enzyme TPH1. So this is data from our lab, where we see that in SPNETs, both from the primary and the liver sites, the TPH1 expression is orders of magnitude greater than the TPH1 expression in normal small bowel tissue. And we've also characterized the TPH1 protein levels with IHC staining in primary SB nets, and we see very high expression of TPH1 in the tumor tissue. The reason that we are interested in serotonin in neuroendocrine tumors, or the reason we're interested in TPH1 is that it is the rate limiting step of peripheral serotonin biosynthesis. So in neuroendocrine cancer cells, so tryptophan is converted to serotonin via TPH1, and then in low-grade tumors, the serotonin is usually metastasized by the liver and excreted in the urine, and can be used as a biomarker and a diagnostic marker for these tumors. But then in later stages when liver metastases develop, serotonin secreted by these tumors can enter the systemic circulation and contribute to the classical symptoms of carcinoid syndrome. And there's been a TPH1 inhibitor, teltristat ethyl, that's been developed that can reduce serotonin production by these tumors and is used to help manage carcinoid syndrome in patients with these small bowel tumors. So although the link between serotonin and carcinoid syndrome has been well established, it's not well understood what the role of serotonin in neuroendocrine tumorigenesis is. And that's what this project is really focused on. And specifically, we're interested in 3 different aspects of tumorigenesis. And one is if there's any effect of serotonin on cell proliferation, as well as effects of serotonin on tumor angiogenesis, and the ability of these tumors to metastasize as they have a strong propensity to metastasize to the liver. So we've taken a genetic approach to study serotonin synthesis inhibition in these tumors, and the model that we use is the Bond cell line, which is actually derived from a pancreatic net, but it has very high TPH1 expression, making it a good model to study serotonin metabolism. So we have generated Bond cell lines with stable knockdown of TPH1 using 2 different shRNAs. And you can see on the right, with Western blotting, we were able to achieve very efficient knockdown of TPH1 in these cell lines. And then we assessed the cellular proliferation of these cells in vitro, as well as the ability to form tumors in vivo. So the results from the study were very interesting. We found that the TPH1 knockdown cells in vitro are not affected by the knockdown, and their cellular proliferation is the same as the parental cells. However, we found that in vivo, knocking down TPH1 significantly reduces tumorigenesis. The tumors are much smaller and grow at a much slower rate. And as you can see with these images here on the bottom, the control tumors are highly vascularized, whereas the knockdown tumors are pale and appear to be significantly less vascularized. And after observing this phenotype with the TPH1 knockdown, we wanted to see if we could recapitulate it with teltristat by pharmacologically inhibiting TPH1 and teltristat. And we were able to achieve a very similar result, where we saw that in vitro, teltristat does not affect Bond cell viability, but in vivo, when we treat mice harboring Bond xenograft tumors with teltristat, we see a significant reduction in the growth, very similar to the effect of TPH1 knockdown. And again, we see that the teltristat treated tumors are less vascularized. So based on what we saw visually with the vascularization of tumors, we wanted to look further into angiogenesis because we felt that this was one of the main mechanisms that serotonin may be affecting tumorigenesis. So we performed CD31 staining, which is a marker of endothelial cells, and we can see here in figure A that the TPH1 knockdown tumors have very little staining and very few microvessels, whereas the control tumors are highly vascularized. And in B, we also see that the expression of two different isoforms of vascular endothelial growth factor, B and C, are significantly reduced in the knockdown tumors compared to the control tumors. And then we were also interested on the effect of serotonin directly on the endothelial cells. So we performed a tube formation assay with HUVEC cells. And in the normal morphology of these cells in culture is they acquire a spindle shape and form cell-to-cell connections. And that's what we see when we culture them with media from the bond, conditioned media from the bond cells. However, when we culture them with conditioned media from the TPH1 knockdown cells, their morphology is more round and they form fewer cell-to-cell connections, indicating there's an impairment in their cellular function. And then interestingly, when we add back serotonin to the TPH1 knockdown media, we see a restoration of the normal cell morphology, indicating that serotonin is important for supporting the endothelial cell function. So returning to this figure, what we learned from these studies is that it doesn't appear that serotonin directly affects cellular proliferation, which has been seen by other groups as well as us. But we found that TPH1 and serotonin is very important for tumorigenesis, as we saw that the TPH1 knockdown tumors are much smaller than the control, and telotristat was able to reduce the growth of the bond xenografts. And we found that one of the main mechanisms by which serotonin is promoting the growth of these neuroendocrine tumors is through increasing angiogenesis by directly affecting the endothelial cells. So although we learned a lot from this study, there's still many remaining questions in terms of the effect of serotonin on tumorigenesis. Serotonin is a very dynamic metabolite and it can be difficult to study. And some of the remaining questions that we want to look into are the role of serotonin at different time points in tumor progression, the effect of the tumor microenvironment on serotonin metabolism, and also if there's any effect of serotonin on net metastasis. So in order to address these questions, we believe that a new method of serotonin detection that could provide more insight into the dynamic nature of this metabolite was needed. So that brings us to the serotonin sensor. So we have been developing a luminescent serotonin sensor for studying serotonin metabolism in real time in neuroendocrine tumor cells. A few years ago, a group from UC San Diego developed the sensor you see in this black box. It consists of a serotonin binding domain termed iSero, and then a green fluorescent protein reporter. And this sensor can be genetically encoded into the genome of a cell line of interest. And we felt that this could be a very powerful tool for studying serotonin metabolism in neuroendocrine tumors. But we specifically wanted the sensor to have a luminescent readout, since many pharmaceutical compounds as well as biological tissue exhibits high levels of autofluorescence. So we developed a new version of the serotonin sensor using the iSero domain and a renylluciferase reporter. So I'll be referring to the sensor as the iSero-RLuc sensor for the rest of the talk. So here on the right is just a schematic of essentially how the sensor works and how we're using it. So we genetically encoded the sensor into the bond cell line, but it theoretically could be used in any cell line of interest. And then the cells express the sensor in the cytoplasm, where it binds, where the iSero domain binds to intracellular serotonin. And by adding exogenous colentrazine, which is the substrate for renylluciferase, and measuring the light produced, we can detect relative levels of intracellular serotonin. So we first characterized this sensor before moving to the bond cell line in budding yeast. So on the right, we constructed two different versions of the sensor, and we found one version when we add exogenous serotonin, we see an increase in the luminescence activity of the sensor. And then over in figure B, we're testing a range of serotonin concentrations, and we see as there's more serotonin in the environment of the sensor, there's a corresponding increase in the luminescent activity of the rhinoluciferase reporter, indicating that the sensor is able to detect serotonin. So then we moved on to the bond cell line model by stably expressing the ICER-Arlic sensor in bond cells and assessing the effect of different growth states on serotonin synthesis. So it's been previously reported in the literature that bond cells grown in the suspension growth state appear to secrete more serotonin than cells grown in a monolayer or adherent growth state. So using the ICER-Arlic sensor, we looked at the serotonin synthesis in these two growth states. And what's really interesting is we found that the serotonin synthesis was substantially increased when cells were grown in the suspension growth state compared to the adherent. And we also performed mass spec analysis to confirm this, that the suspension cells contain more intracellular serotonin. So this was a really interesting finding, that not only are the cells secreting more serotonin when they're in the suspension growth state, they're actually upregulating serotonin synthesis and storing more serotonin intracellularly. So then we want to look at different growth conditions to see how the extracellular environment of the sensor may be affecting serotonin biosynthesis. So what we found is that serum starvation actually induces serotonin biosynthesis. So in figure A, we're comparing the standard 10% FBS media to cells that were cultured in media without FBS. So we serum starved them for 24 hours. And we found that induced serotonin synthesis based on the increase in the sensor activity that we observed. And then in B, we were looking at different concentrations of FBS compared to the control 10% FBS condition for 24 hours. And we see as there's less FBS in the media, there's a corresponding increase in the activity of the sensor, indicating that more serotonin is being produced. And then in C, we're interested in what time point this change occurs. The first two experiments were both done at 24 hours. But in this experiment, we found that at just half an hour of serum starvation, we already see a big increase in the sensor activity, indicating that serotonin synthesis is rapidly upregulated upon serum starvation. And we saw a peak at 2 hours, and then it tapered off, but it's still upregulated at 24 hours. So based on this experiment and the fact that we saw an increase in serotonin at just half an hour, we predicted that it was likely a post-translational modification of TPH1. And it's been previously reported in the literature by Banik et al. That TPH1 can be phosphorylated at serine 58, which relies on its regulatory domain. When TPH1 is phosphorylated, it has significantly higher enzymatic activity. And so we performed Western blotting with a phospho-specific TPH1 antibody. And what we see is that under serum starvation, there's an increase in the levels of phosphorylated TPH1, whereas the total level of TPH1 enzyme remains the same. Then we also looked at, as in the previous slide, different concentrations of FBS, and we see as there's less FBS supplementation in media, there's an increase in the amount of phosphorylated TPH1, and again, the total TPH1 remains constant. And then we looked at different time points, and we see just as in the serotonin sensor assay, at the half-hour time point, there's a big increase in the levels of phosphorylated TPH1, which is then maintained over time. So from these experiments, we think that phosphorylation of TPH1 under serum starvation is what is leading to an increase in serotonin biosynthesis. So this leads to the current working model that we have for serotonin synthesis regulation in the tumor microenvironment. So in the tumor microenvironment, there is a nutrient gradient depending on how far the tumor cells are from the vasculature. The cells close to the vasculature, which have an ample supply of nutrients, are highly proliferative. And then as you move away from the vasculature, the cells have fewer nutrients and are less proliferative. And at the furthest point, there are necrotic cells where there's very low nutrients and the cells are either not dividing at all or are dying. So what we observed with the serotonin sensor, we believe that these cells in the intermediary area of the tumor where there's low nutrients, but yet there's still viable tumor cells, but just not rapidly dividing, are the cells that are upregulating serotonin synthesis, which that serotonin is then signaling to the endothelial cells to promote angiogenesis into new tumor environments. And we're currently working on quantifying IHC staining that we've done for TPH1 and serotonin on a group of patient tumor sections that we have. So we're going to look to see how this model fits into what we see in the patient tumors. And then the second part of this model, based on what we observed with our anchorage-independent growth studies, we believe that when the tumor cells escape into the vasculature, they're upregulating serotonin synthesis since they're in an anchorage-independent growth state. And we're predicting that that increase in serotonin may somehow aid in metastasis, which is an area of research that we're interested in looking into. So with that, I would like to thank my research mentors, Dr. Ear and Dr. Howe, as well as everyone else that was involved in this work. And I would like to thank everyone for listening, and I welcome any questions.
Wonderful. Great job, Dane. Anchorage, what do you think? Do you think we can do 2 or 3 minutes of questions before we move on?
Yeah, I think we can go for a few questions. Yeah. So, anyone have questions? So maybe I will start with a question. So this is quite interesting, Dan. And I actually am a co-author in that. Thanks to Tow for this. Maybe I should have read this before, but is the model that you used, mouse model, is it orthotopic or is it subcutaneous?
We used a subcutaneous model.
Because the reason I'm asking is because you showed in vitro, the results were different from in vivo. So I'm just wondering, would there be any difference between the subcutaneous versus the orthotopic?
Yeah.
How you want to answer?
Poe has.
Yeah.
Well, Dane can go first, or I can continue, or if you want me to answer, I can address that too.
Yeah. I would say I think the difference that we observe between the in vitro and the in vivo state is that in vitro, the cells are really in like an ideal growth state where they don't have to compete for nutrients. And so I think when we inhibit serotonin synthesis. The serotonin doesn't have an autocrine effect on cell growth. I think the cells are growing as fast as they can with the available nutrients in vitro. But then in vivo, the cells have to compete for nutrients, and it appears that the serotonin is actually promoting angiogenesis to the tumor to enable more nutrients. But specifically, I'm not sure quite what the difference between sub-Q and orthotopic model is. So I may have Dr. Ear addressed that part of the question.
Yeah, so I just want to point out that we do have data Dane presented here on the subcutaneous tumor model. We also have data on a liver metastases model, where when we actually inhibit the serotonin synthesis with the inhibitors telotristat, we also see a growth reduction. And so that's why we think that I would think that the mechanism is quite conserved for solid tumors. We also actually there was a very interesting clinical paper from Dr. Metz's group, like I think in the past years, that actually is on patient data. So they looked at liver metastases, and they quantify liver metastases from patient with and without telotristat ethyl treatment, and they found in their cohort that there's actually decreased growth with telotristat treatment. So I think there's room for more study at the moment. There's very few groups that are looking at this at the moment, because there was previous data that were published from in vitro data that showed that there was no difference. But I think, you know, a tumor in the tumor microenvironment in the patient and is quite different from the culture dish. So I think we should definitely focus more on the in vivo model or patient data looking forward.
Definitely, it makes sense, because you're going in the angiogenesis angle, then it will not make sense in vitro as well. Any other question?
Right, I think we're gonna have to move on to our next speaker. So, Angaraj.
Yes. The next speaker is Katie English. Title for this talk is DNA hydroxymethylation in pancreatic neuroendocrine tumors. Katie is an endocrinologist, where she completed her clinical training in Brisbane, Australia. She is currently in the final year of her PhD with Professor Raj Thakur and Dr. Kate Lyons at the University of Oxford, looking at DNA modifications in pancreatic neuroendocrine tumors. The DNA modification she's studying is called 5-hydroxymethylation, and this epigenetic mark can change how DNA is read by the cell, and it's important for normal functioning of cells. Her work has mainly focused on this dysregulation, which is seen in pancreatic neuroendocrine tumors, and how modulating this DNA modification with drugs may help to improve cancer therapies. Without further delay, welcome Katie to give this talk.
Okay, so hopefully, can you see my screen?
Yes.
We can, but I'm seeing like the presenter view.
Ah, okay, let me try.
There you go. Perfect.
Excellent. Okay, thank you for the very kind introduction, and thank you for allowing me to speak at the NET Cancer Foundation Research Summit today. So yes, I am a PhD student at Somerville College here, and I'm very lucky to have three supervisors, Professor Thacker here and Dr. Kate Lyons, and Eric O'Neill, who's at the Oncology Centre here. So basically, I'm in my final year, and I've been looking at DNA hydroxymethylation in pancreatic neuroendocrine tumours. So what I thought, I know we've had a bit of chat about epigenetics, particularly with DNA methylation, but I thought I might just do a little bit of a background first and then discuss briefly about what I've been doing. So very briefly, the pancreas is found in the upper abdomen, and it's mainly responsible for two things. So we either have exocrine pancreas that is responsible for secreting enzymes that mainly break down food, and that's the majority of the pancreas. However, the one we're particularly interested in is the endocrine pancreas, and that predominantly secretes hormones, which mainly regulates blood glucose, although it does secrete hormones that do some other things as well. So where the islets are, so they're basically called islets of Langerhans, and the majority are made up of beta cells, so it's approximately half of the cells, and they secrete insulin. The other cells in the pancreas are your alpha cell that secretes glucagon, which is sort of works as a counter-regulatory hormone to insulin, as does the delta cell somatostatin. So pancreatic neuroendocrine tumours, they either arise as part of a hereditary tumour syndrome, so that would be a germline mutation, for example, MEN1 syndrome, or they can arise sporadically, so this essentially means that there's no cancer predisposition syndrome. So most patients with MEN1 syndrome have a germline mutation in MEN1, which is a tumour suppressor gene that encodes for the protein menin. And quite a lot, so up to 80% of these patients with these germline mutations develop pancreatic neuroendocrine tumours at some point during their lifetime. However, in sporadic PNETs, the most common genetic mutation still is in the MEN1 gene. However, this is only present in approximately 40% of patients. So I've just put on the right-hand side, this is the normal protein structure of menin, so that's in grey. It's a 610 amino acid structure. And what you can see in the tiny red squiggle sort of in the pocket of menin is the MLL protein. So that is one of the proteins that interacts with menin. So menin typically itself doesn't interact with DNA, although it interacts with lots of other proteins to form a complex, which is involved in particularly epigenetic regulation. So what I've put down the bottom for some of our viewers who may not be as up to date or with what an MEN mutation might look like, you might be able to see a tiny little blue section in the protein. So that is an example of a mutation. And now, because that amino acid has changed, the red squiggle or the MLL protein is unable to bind to the menin protein. So therefore, functionally, it can't work anymore. And that's one of the mechanisms by which MEN mutations can lead to a loss of function and to a tumour. So then the question arises, why, if we only have 40% of patients have an MEN1 gene mutation? Yes, there are other gene mutations associated with PNETs, but the majority still, we don't really know why they form. So then the question arises, could it be something to do with the epigenome? So that is what I wanted to have a look at. So then what is epigenetics? Well, it basically refers to a process that alters gene activity without changing the DNA sequence itself. So we know that DNA is quite long, so it's almost up to 2 meters in length, and it's packaged tightly within the nucleus around histone proteins, which are the building blocks of nucleosomes to ultimately form chromatin. So epigenetic modifications can include DNA methylation marks and or chromatin marks, which mark the DNA and will change this structure to either open or to closed chromatin. So open where transcription is allowed to happen, or closed where it's not. So on the right-hand side is a schematic. So you see the chromosome in purple, and it's wrapped tightly around these, they almost look like macaroons. They're yellow on the diagram, and they have histone tails, so lots of little squiggles that come out from the histone that can be modified with lots of different marks. For example, methylation, sumylation, phosphorylation, and all of these different marks on different histone tails will lead to a different outcome with the chromatin state. So for example, menin comes back in here. So on the top right-hand side, menin leads to the formation of the open chromatin mark. So this is H3K4 trimethylation. And the MLL1 enzyme or protein is involved with this, so putting on this mark. In between those two histones, you can see an open section of chromatin, which has blue circles, which is the unmethylated DNA. So essentially, DNA methylation and histone marks tend to go hand in hand. So you have open chromatin with an with unmethylated DNA. So we used to think that DNA methylation was relatively static. You'd say, okay, we have DNA methylation, therefore there is no transcription, and you have closed chromatin. Well, we now know that's not entirely true. So the DNA methylome is dynamic. You have enzymes that remove methylation, and we have enzymes that put the methylation back on. And this doesn't specifically have to happen between replication. You can actually just take methylation marks on and off on unreplicated DNA. I'll just draw your attention down to the bottom of the diagram. That's an example of heterochromatin or closed chromatin. So you have DNA methylation associated with one of the histone marks, which is associated with closed chromatin, which is on one of the different histone tails. So this is an H3K27 trimethylation, as opposed to the H3K4 trimethylation, which is associated with open chromatin. So having a little looking deeper at this DNA hydroxymethylation, menin's also associated with changes in DNA methylation. So what we know is if you lose menin, it leads to DNMT, so this is DNA methyltransferase, which is responsible for putting on the methylation mark to the 5' position of cytosine, in addition to the open chromatin mark, H3K4NE3. And what you can see as you go round the circle, there's a newly discovered enzyme. So there are 3. There's the 1011 translocase enzymes that can progressively take this methylation mark off. So it goes through different marks. You have 5-hydroxymethylcytosine, so that's the next mark along. And interestingly, this mark is actually associated with transcription. So it's different to 5-methylcytosine, which is associated with silencing transcription. So it's a circle. So essentially the 1011 translocase enzyme can keep taking off the mark through subsequent reactions back to an unmethylated cytosine, which then may or may not become methylated again with DNA methyltransferase enzymes. So 1011 translocase is an enzyme which is dependent on 2 things. It needs iron, so Fe2+ in the serum, and it also needs alpha-ketoglutarate, which is part of the citric acid cycle. So this nicely links in cell metabolism with the DNA methylome. So I'm going to keep going. So what might change to do with cancer? So we know that there are DNA methylation changes or dysregulation with cancer. So essentially genes that are not meant to be methylated become methylated, which leads to transcriptional repression. And overall, however, cancer cells are globally hypomethylated. So we have global hypomethylation with inappropriate specific DNA methylation. Now, interestingly, recently it's shown that at promoters, mainly promoters, you have protected areas called CPG sites. So this is areas of DNA where there is a cluster of cytosine followed by guanine on the same strand of DNA. So throughout the whole genome, we actually find that there's a less than expected frequency of these CPG sites, and it's thought to be due to spontaneous deamination to thymine over time. However, we find about 30,000 of these sort of pockets that we call CPG islands that have a greater than expected frequency of CG sites. And interestingly, they tend to be around the promoters of housekeeper genes. And also we find that the 5-hydroxymethylcytosine mark tends to be on the shores, so on either side of the CPG, with the thought that this protects the unmethylated cytosines in the middle of the CPG island from DNA methyltransferase enzymes. However, a cancer cell, which I'll discuss later, can lose this hydroxymethylation. Therefore, there's nothing now to protect the unmethylated cytosines in the middle of the CpG island, and the DNA methyltransferase enzyme will subsequently put on this methylation mark and lead to silencing of transcription. So I wanted to investigate the DNA hydroxymethylation in tumour samples from patients with PNETs. And why I found this interesting is that current techniques aren't able to differentiate between DNA methylation and DNA hydroxymethylation. So this is really important because if one of these methylation marks is associated with gene repression and the other one is associated with gene transcription, we're sort of inadvertently saying that a lot of CpG sites and or promoters are actually methylated when they're not. And this may be the reason why we have discrepancies with reported methylated genes that are actually expressed. So first of all, we were very lucky enough to have some patient samples. So we have 70 samples that were first characterized. So these were mainly non-functioning PNETs and insulinomas. So on the left-hand side, there's the hematoxin and eosin staining for a non-functioning PNET, which is the first column, insulinoma the second, and their adjacent normal islets in the third. So we stain them for H&E synaptophysin as a marker of a neuroendocrine tumour and for menin. So next, given the fact that PNETs are relatively rare tumours and they're quite difficult to come by, a lot of the samples were up to sort of 30 years old. So these were preserved in wax blocks. So therefore, it was essential that the tumours that we studied had to have adjacent normal tissue on the same slide to enable comparison. So what we ended up with was 17 insulinomas and 16 non-functioning PNETs. So the first thing that I did was look at what the DNA 5-hydroxymethylcytosine mark was in normal adjacent islets compared to their tumours. And what you can see on the top panel, so there's the normal exocrine tissue, which looks like a four-leafed clover, and an arrowhead for the normal islet. And what you can see is there's quite heavily staining. So this is a dab staining, which essentially the darker the stain of brown, the more highly expressed the thing of interest is you're looking at. So you can see incredibly brown nuclei in the normal islet compared to the adjacent exocrine tissue. And in the tumour cell, you can see that there's really not much DNA hydroxymethylation at all. So you've lost the hydroxymethylation in the tumour. So this was subsequently carried out on the remaining samples. And on the right-hand side, what you can see is that there is a significant decrease in DNA hydroxymethylation compared to their adjacent normal islets. And essentially, it's the same as the exocrine tissue that you can look at. So then the next question was, okay, so you've determined that there's global changes in DNA hydroxymethylation. What about the specific CpG sites themselves? So what I did was I extracted DNA from PNETs and normal adjacent tissue from patient samples. So I had 4 insulinomas, 5 non-functioning PNETs, and 7 normal adjacent tissues. So this was 4 adjacent tissue, sorry, and 3 separate normal pancreas that we obtained from the Oxford Research Biobank, and that underwent methylation epic array to determine specific DNA hydroxymethylation. So what I did, I used a slightly different technique for this because this is a methylation epic array. So for each sample of DNA, I split it into 2, and one of these paired samples underwent an oxidative step first to change the 5-hydroxymethylcytosines further along in the cycle to a 5-formyl cytosine or 5-carboxy cytosine, and that actually gets converted with bisulfite. So then by looking at the difference between the 2 samples, I could determine the DNA hydroxymethylation of specific CpG sites. So I haven't shown the first bit of data, but essentially what it shows is that normal pancreatic tissue has significantly higher amounts of DNA hydroxymethylcytosine. So it's 8% of the more than 8 800,000 CPG sites that were investigated, compared to less than 1% from the pancreatic neuroendocrine tumours. So there's a significant decrease in DNA hydroxymethylcytosine, which is not unexpected, because it is in keeping with what we found by immunohistochemistry. Then what I wanted to do is to look to see what pathways might be important for these DNA hydroxymethylcytosine. And what you can see on the right-hand side are the different KEGG pathways that came up. So this was using a false discovery rate of less than 0.05. And interestingly, some of the pathways involved particularly were glucagon signaling, TGF-beta signaling, Notch signaling, and also the insulin signaling pathways. So basically, in summary, what I found, or what I've shown today, is that normal islets are heavily hydroxymethylated, shown by immunohistochemistry, and which may protect important pathways from being methylated. One of the unanswered questions at the moment are, why do islets have such a high degree of DNA hydroxymethylation when compared to other tissues? And then that leads on to the next question, that when pancreatic neuroendocrine tumours lose DNA hydroxymethylation, is, what happens? Are we able to alter this DNA modification? Can we put it back on? And if we can put it back on, is this actually beneficial? The other question that's unanswered is, we know that menin is associated with DNA methylation, but what is its association with DNA hydroxymethylation? And these are the questions that I am currently working on, and hopefully we'll have some answers to that shortly. And basically, this is my acknowledgement slide. These are all the people that I've worked with in our lab together. So hopefully you can see that that spells out NET. So that is, we have Kate Lyons as the N, and I'm the E in the middle, and Omer Sharik is our T, and he's one of the other DPhil students that I had the pleasure of working with when he had his time here. And also absolutely to all the patients who've donated their samples, and to Cancer Research UK, and to my college here, Somerville College at the University of Oxford. And thank you for letting me present my data in your research summit.
Thank you, Katie. I think with the interest of time, Rachel, do you think we should move on to the next talk, or do we have time for a question?
For the interest of time, we are going to move on to our next.
Right. Okay. Thanks, Katie.
That's right. Thank you. Okay.
Right. Thank you so much again, Katie, for speaking to us. And so to move forward with this conference, I am going to introduce our next and final speaker. So we are very excited to have Dr. Dawn Quelle here. She is a professor of neuroscience and pharmacology and pathology at the University of Iowa, Carver College of Medicine. Dr. Quelle earned her PhD from Pennsylvania State University, where she studied cytokine receptor signaling, and then she did her postdoc research at St. Jude's Children's Hospital, where she established the role of cyclin D-dependent kinases in promoting cell cycle progression, and discovered the ARF, or alternative reading frame tumor suppressor, while cloning the murine INK4A/ARF CDKN2 locus. She then joined faculty at the University of Iowa in 1997 and has led the Cancer's Genes and Pathways program in the Holden Comprehensive Cancer Center since 2009. Currently, her laboratory studies mechanisms of tumor pathogenesis with a focus on neuroendocrine tumors, and her research is funded by multiple PI grants from the NIH. Her team is working tirelessly to translate their discoveries into new treatment options for patients with neuroendocrine tumors. So without further ado, I would like to introduce Dr. Dawn Quelle.
Thank you so much, Rachel. I hope I don't get a frog in my throat while I'm talking. But thank you all for the invitation, and what a nice job everybody has done before me, especially Dane and Katie. I love seeing all the great work by the trainees. So I will try and share my screen here. Here we go. So can you see my, let's get this in presenter mode. You probably can't see my presenter mode, can you? Hold on a minute. Yeah, I have it on the wrong monitor, so I just have to change my monitor.
Yeah, take your time.
Should have done this before. Let's see. If I get this going right now, I will move you guys over here, and that way I can see it over here. Okay, there we go. I think we've got it. Hey.
It's usually how I am with my one laptop. I've tried 3 things. All right, looks great, Dawn.
All right, wonderful. As I finally get going, how many times have we given presentations like this? And each one is a new experience. Okay, well, thank you all. Hopefully you can hear me just fine too. So I really thank you for this opportunity to tell you about our work, which I hope you'll agree by the end of my talk, there is value in trying to target multiple kinases, such as CDK4/6 and MEK kinases, to potentially alter the immune environment and promote immune therapy for pancreatic NETs. But I think this therapy could be very useful in not only targeting the primary, but also potentially metastatic lesions. So we all know that neuroendocrine tumors are a really challenging disease. The incidence is rising quite precipitously. And I just pulled together some incidents from the surveillance and epidemiology and end results data set, which covers a lot of US tumors from the 1970s up till now. And you can see that there's a tremendous rise from about 2 per 100,000 up to over 8. So a more than 4-fold increase in incidence. Now, these are slow growing tumors. They don't respond well to traditional chemotherapies, and we don't really adequately understand all of the drivers that cause this disease to form. So my group has been focused primarily on pancreatic NETs and just trying to understand their biology better and identify new treatments. So there are a number of good treatments already for these cancers. And so we can target somatostatin receptors, AKT-mTOR signaling, receptor tyrosine kinases. But in those patients who have unresectable disease, sadly, those will inevitably progress. And that is where we're left with a problem, because we need to know what can we treat those patients with. We need to have better targets and perhaps better combination therapies for treating the disease. So on the left here, you can see this cartoon of different pathways that are dysregulated in pancreatic neuroendocrine tumors, and many of them are druggable. And so what I'm going to tell you about is our strategy for targeting CDK4/6 and MEK kinases is based on the fact that they are dysregulated in these cancers. You know, we know that CDK4 and 6 are upregulated in the tumors, that there is loss of inhibitors of these kinases like P16, and that connotes poor prognosis in the patients. We see upregulation of factors that turn on these kinases like Rabl6A, which my lab studies, and also MEK is upstream of CDK4 and 6. We have performed some global kinome analyses, which really just look at what are all the kinases that are activated in neuroendocrine tumors. And we compared growing versus non-growing PNETs, and we found that there are actually 16 really highly reproducible kinases that are up in the growing cells, and those include cyclin-dependent kinases 6 and 16. We also conducted phosphoproteome analyses on these tumors, growing or not, and in the growing tumor cells, we see that there is hyperactivated CDK4/6 and also MEK. So all of this data really pulls together to justify our focus on targeting them in the cancers. And we also want to mention that, you know, there have been several clinical trials in PNET patients where they have attempted to inhibit CDK4 and 6 with monotherapies. While the trials were successful in being conducted, they were not successful in showing any benefit. So what can we do to improve that? And what we hypothesize is that we need combination therapy. and that we can target both CDK4 and 6 as well as MEK, and that ideally that would have really good synergistic activity at suppressing the tumors. And the two people who led this in my lab are wonderful, talented research scientists, Salma and Courtney. So there were two beautiful papers in Science and Cell from Scott Lowe's group that actually showed you do get synergy by combined targeting of CDK4/6 if you use a kinase inhibitor like palbociclib, which directly binds the kinase and blocks its activity. And if you move upstream and you target MEK kinases, they use trametinib. In our studies, we use mirdametinib. But if you target both of them, MEK inhibition blocks the ability of MYC to transcribe and increase the expression of the kinase subunits. So you really have a double whammy effect, and you block the phosphorylation of RB, the retinoblastoma tumor suppressor, and you hyperactivate it. And that leads to tumor stasis through increased senescence. And the idea is that you can also modulate immune infiltration and activation of T cells in the tumors. So we looked at this combination in BON and QGP1 cells, and you can see that there's beautiful synergy at lower doses of both of the drugs. So then we wanted to validate it at the molecular level, and you can see that we have reactivation of RB as measured by the decreased phosphorylation of it when you treat with the combination as opposed to the individual drugs. Also, we see increased cell cycle arrest with the combination shown in red compared to individual drugs or vehicle, and increased cell death. So that was good in vitro data. Those are really quick assays. So we moved to longer term colony formation assays, which take 2 to 3 weeks. And we also saw that the combination led to not only fewer colonies, which is shown in red compared to the controls, but also smaller colonies. So that was pretty exciting, but still we're in vitro. So we moved to mice. And what we did was we put BON1 cells. These are just subcutaneous xenografts, and they're in NSG mice that are immune deficient. But we were excited to see that we even had some effect of palbociclib and mirdametinib individually at suppressing tumor growth compared to vehicle, which is shown in the black curve. But when you use the combination shown in the red curve, we had a 6-fold extension in survival, which was pretty impressive and unexpected because we are in immune deficient mice. So while that was really good, we also never saw tumor regression. We only slowed the growth. And eventually all of those tumors grew back. So as the rest of my talk, I'm going to tell you about a metastasis model and also tell you about getting into immune competent mice. So in this metastasis model, we basically have luciferase cells. So they express luciferase in BON and QGP1 cells. We inject them into the tail vein, and they will form tumors throughout the animals. And we can monitor the tumor growth by bioluminescence imaging. Courtney has done a lot of these studies along with Salma. And on the left, you can see that if we use the combination shown in the pink curve, there is slower metastatic tumor growth over time compared to the vehicle and the palbociclib treatments. We weren't quite significantly different from the mirdametinib alone, but if we went longer, we do see the differences. And you'll notice that really in the combination and the images of the mice below, we see very little tumor burden. And impressively, we actually reduce the number of tissues that are colonized by those tumor cells. So we really block that. So that's a suggestion that we could impair metastatic spread of the tumors with this combination. But we wanted to get into immune competent cells. And so we worked with our friends at Rutgers, Steve Labuti, Ned Latame, and especially Xi Zhang, who carried out the experiments. He developed a MEN1-PTEN double knockout animal with a PDX1 Cree driven knockout of those genes, and they develop insulinomas in about 5 to 6 months. And you can see on the left that they actually express much higher levels of CDK4 compared to MEN1 single knockout tumors. So that also, was an impetus to say maybe they'll be more sensitive to targeting CDK4 and 6. So the idea for our experiment was to treat the animals for 2 weeks with either vehicle, MEK inhibitor alone, CDK4/6 inhibitor alone, or the combination. And we predicted, and then we would harvest the tumors 4 weeks later. We predicted that we'd have the smallest tumors with the combination. So that was the case. You can see the H&E images of the tumors as well as the Ki-67 levels. And the tumors got much smaller with the combination, and that was just quantified over here. So we saw dramatic tumor regression. We wanted to look at what happened to the immune cell infiltrates in the tumors. And impressively, in the red, you can see the combination. We saw an increase in B cells and plasma cells, as marked by CD19 on their surface, as well as antigen-presenting dendritic cells. So we were really excited by this. And that was especially the case because tumor-infiltrating B and plasma cells in other human tumors are associated with better patient survival and increased formation of these structures called tertiary lymphoid structures that are kind of like lymph nodes within the tumors. And they contain all of the relevant immune cells so that you can have anti-tumor immune destruction. And in fact, the presence of B and plasma cells is associated with increased efficacy of immune checkpoint inhibitor therapy in patients, either with anti-PD-1 or anti-PD-L1 therapies. So we predicted that we would actually induce a better response to immune checkpoint inhibitor therapy with this combination of CDK4 and MEK inhibitors. Now, we wanted to actually just use one of the kinase inhibitors, palbociclib, because that would be more immediately translational to patients. So in this experiment, we compared vehicle and an IgG control versus palbociclib and IgG. And you can see that in the gray, the palbociclib actually has a pretty good ability to decrease the serum insulin, suggesting reduced tumor size. And you can see that in the gray bar. It did have a pretty good effect on its own. PD-L1 therapy, targeting that immune checkpoint inhibitor, was pretty good, also on its own. But if you use the combination shown in blue of both CDK4 targeting and PD-L1, you had synergistic effects. So at this point, I have just a few more slides after this, but we know that this combination therapy synergistically suppresses PNETs in vitro and in vivo. It works better in immune competent mice and is associated with a B or plasma cell infiltration. And the combination sensitized the tumors to immunotherapy. So we want to expand those insulinoma therapy studies. We want to do more studies in immune competent mice that have non-functional PNETs, and through that working with Rachel and others at University of Alabama with their model. And then we want to test the importance of the B in the plasma cells. But just to finish off, I want to tell you we've made a little progress at looking at the tumor infiltrating immune cells by flow, cytometry, and multiplex immunohistochemistry. So here's the flow. Zhejiang did this, and you can see that in the combination treated animals, there's a trend. We don't have a lot of animals yet, but there's a trend toward increasing plasmablasts in those tumors, which is what we would predict, and also dendritic cells. But noticeably, we did not see an increase in CD4 or CD8 positive T cells. But what we wonder is whether they're aggregating and forming tertiary lymphoid structures. So to do that, we want to do multiplex immunohistochemistry. And this is basically an approach where you do a stain on a single slide, you get an image, you scan it, and then you destain, and you do it over and over again. So iterative cycles of staining, destaining, and imaging. And we're working with the Knight Cancer Institute at the Oregon Health Sciences University. They've been doing this for 15 years, and they are expensive, but it is working. And so what we have done is we took a 10-year-old patient specimen and a 2-year-old patient specimen and from our archives. And basically, you can see that the multiplex IHC worked beautifully on both, even the very old tumor samples. We're seeing a lot of CD8 positive T cells in green, but we're seeing a wide variety of immune cells in both of these tumor specimens, which kind of matches some of the data that are out there, although there isn't much known about immune composition in these cells. We didn't study the plasma cells because they didn't include a marker for that, but they do have another staining panel that will really give us a great breakdown of B cells, plasmablasts, plasma cells, etc. And that's what we want to subject a lot of our mouse tumors and our human tumors to evaluate their immune composition. So with that, I want to thank everybody for your attention. And, you know, my lab is small, but they're wonderful people. Courtney and Salma were especially leaders in this. And I've got just an amazing group of collaborators here at Iowa, including Poe, and then collaborators at Rutgers. So thank you very much for your attention. And I will stop. Let's see, I will stop sharing.
Oh, wonderful. Thank you so much, Dawn, for that excellent presentation. We do have a couple minutes for questions. So does anyone have questions at this time for Dr. Dawn Quelle?
It was like a data deluge, wasn't it? Really fast. Sorry about that.
No, it was great, Dawn. I think you guys have really done comprehensive work in vitro and in vivo, which is really nice to see. And especially, you know, integrating the immunocompetent models is very exciting work.
We really are thankful to all of our colleagues, basically, because we all know it's hard to find good models to do the studies in. And what I love from the earlier talks, too, is just the number of patient specimens that people have been evaluating. What I wish is that we would be able to congregate and basically pool all those precious samples with all the data associated with them. I know we've been talking about that with models consortium groups and so forth, but being able to share that data with each other and share the samples, I hope we can get there.
I absolutely agree. And Garaj.
Yeah, Dawn, this is a really amazing talk. It's very interesting, and it's essential given the neuroendocrine tumor and immunotherapy. So I'm just, related to your point about, bringing to these models. So I'm just curious, did you do this model with the genetically engineered mouse model, or is it like a cell line from the model that you injected?
Right. It was all done in the mice, and it was our colleagues, basically, Zhizheng Wan did all of the studies at Rutgers. But yeah, I think they have some cell lines that if you wanted to actually work in the cell lines and maybe run lots of tests of different drugs against them and so forth to sort of pilot, what do I want to do in the animal? That would be a great idea.
Yeah, that's great. Yeah, I'll contact you. That's yeah. Thank you.
You're welcome.
All right. Do we have any other questions before we wrap up this wonderful conference?
I just want to say thanks again for organizing it. I think that having these conferences more frequently gives people a chance when they can get together just to hear stuff and maybe get new ideas or build new collaborations. So this is one.
Yeah, I have had a bunch of ideas. Oh, Dane's in the back. You can see him, but I've written down all of I've got a page worth of ideas. So awesome. I think this conference has been very inspiring. And I always feel very inspired to keep going when I'm surrounded by all of my colleagues and the patients that attended today, that we're all dedicated to studying this disease.
Yes.
So, all right, with that being said, Selena, would you like to say a couple words before we end?
Right, so thank you to everyone who's attended today, especially to our speakers for giving their time and sharing your research. And I hope you've enjoyed it as much as we have. I just want to say a very special thank you to Rachel and Angaraj for being our presenters today. They've done a fantastic job. Thank you very much. And also just to welcome you to our monthly Net Chatter meetings. They are on the last Thursday of the month, next one being on Thursday, the 29th of June. Heidi, do you want to say who our speaker is for June? I haven't written that down, I'm sorry.
Oh, for June, we invited Dr. Jonathan Hernandez from the NIH. Yeah.
Okay, thank you very much. So thank you all again, and we look forward to seeing you all again soon. Thank you very much again to our speakers. You've done a fantastic job. Thank you.
All right, thank you all. Enjoy your weekends.
Thanks. Bye-bye.
Thank you.